The purified high-quality DNA is ready to use for a wide variety of downstream applications, such as multiplex PCR, coupled in vitro transcription or translation systems, transfection, and sequencing reactions. The importance of the purification of DNA when it's in an agarose gel is to remove impurities or another substance like salts or another DNA fragments that may contaminate the fragment of interest. To purify DNA fragments which have a molecular weight less than 1 kilobase, there are many column extraction kits that could be useful because they ensure a recovery percentage up to 95%, with DNA which molecular weight is between 40 base pairs and 10 kilobase. On the other hand, to purify high molecular weight DNA samples, the most useful method should be the agarose degradation with agarase enzyme, because it is the most recommended for high molecular weight samples.
First of all, a mix was prepared in order to run it in a PCR, adding the remaining of the genomic material and then 5 microliters of loading buffer were added in order to do the mix and then load the well in the electrophoresis gel. The well in the gel was loaded with the mix previously prepared. After this, the electrophoresis was run, and then the band in the electrophoresis gel was cut with help of a UV light. The mass of the gel with the band was measured. Following the method, the buffer L3 was added. In order to melt the agarose gel and to obtain the genetic material, the sample was induced in a water bath. To obtain the genetic material, different columns and tubes were used. 850 microliters were added to the silicon column, and then it was centrifuged at 12,000 gravities. This procedure was repeated twice. The second time of the previous protocol was made. Following the procedure, 500 microliters of wash buffer 1 were added, and the sample was centrifuged again for 3 minutes at 12,000 gravities. Once the filter remaining was eliminated, 50 microliters of buffer E5 were added in order to elute the sample. Previous sample was centrifuged at 12,000 gravities for 1 minute, and then it was quantified with the spectrophotometer, preparing 1 milliliter of dilution 1 to 100 and the blank, that is 1 milliliter of water. The sample and the blank were put in spectrophotometer cells in order to make the read on the equipment. At running the first electrophoresis gel, we can see the DNA band and then some other bloods that can affect the running of the sample. Doing the reads with the spectrophotometers, a low concentration and a negative radio were obtained. Finally, one last electrophoresis was run in order to obtain the purified product of the samples used in the method.
The purity of the samples before and after purification obtained from the 8:60 and 8:80 ratio are very different. The DNA before purification has a slightly lower amount than 1.7, meaning the protein contamination present and the DNA most likely is good quality DNA. The purity obtained after purification indicates that there were problems, because the ratio was a negative one, which can indicate a large amount of contamination by proteins, but was seen by the electrophoresis. Also, there could be that DNA sample was very little. The purification dropped considerably to a 7.8%, which is a low percentage of purification that indicates that the sample has a low probability of working as desired. This may be caused by the insolubilization of the sample, caused by the large amount of agarose with the DNA sample. The UV radiation also could have been more than necessary, causing a damage to the DNA sample.
In the electrophoresis yield, we can see the bands of the DNA approximately at 500 base pairs, and also in the wall number 5 we can see a dark line that comes from the top to the bottom of the gel. That means that the well was damaged while pipetting. DNA extraction with a silica column allows the extraction of the desired DNA fragment only, though it is useful when needing high specificity for further analysis, and it is important to pay special attention to the critical steps to make a successful extraction. For example, the UV light exposure, the use of the correct agarose amount, the correct selection of elution buffer, the correctly wash the column after the elution, and some other steps. All the previously mentioned steps should be followed properly to obtain a higher purification yield and the higher concentration of the DNA possible.